a shy, curious student at Banaras Hindu University, his imagination fired by the mysterious workings of the human brain and the cycles of life all around him. That spark would, for over 2 decades, ignite a journey that transcends borders, disciplines, and even the darkness of a global pandemic. A journey embodied by Dr. Priyoneel Basu. From the bustling labs of Varanasi to the cutting edge neuroscience corridors in Alberta and back to the heart of Odisha, Dr. Basu's story is woven in scientific rigor and genuine human warmth. Dr. Basu's story is woven in threads of scientific rigor and genuine human warmth. While most see science as a pursuit of facts, for Dr. Basu it is deeply personal, rooted in a passion for unraveling mysteries that touch every life.
Research in neuroscience, especially upon how light, sleep, and daily rhythm shape your mind and body, opened new doors in understanding and healing. He was not just a thinker, he was a doer. During the influenza pandemic, he lent his skills to the testing labs where he worked on the front line, ensuring safety and clarity in the time of confusion and fear. As the former dean of KISS Odisha and later a research leader and director, he championed opportunities for many young minds, advocated for science that speaks the language of people, breaking it down into multilingual concepts, making it accessible to every learner. His journey is studied with honor, but the INSA Young Scientist Medal from the National Science Academy stand out. This showcasing his dedication and curiosity.
But it's not just the milestone that makes Dr. Basu unique. Colleagues and students recall his infectious energy, his friendly and jolly nature, and that unmistakable gleam in his eyes when discussing breakthroughs or even teaching the next generation. Even outside the lab, you might find him riding his beloved bike, reminding us that the best scientists are at heart explorers. Dr. Basu's life is a beautiful paradox of exact science and exuberant humanity. This documentary invites you to not just witness his achievements, but to feel the rhythm of his calling, a rhythm that pulses in every experiment, every student's triumph, and every moment of hope he brings to science and life. Meet Dr. Priyoneel Basu sir, a dedicated researcher from our Department of Zoology whose journey through PhD and beyond has shaped groundbreaking work in behavioral neuroscience and circadian physiology.
Hello, I'm Dr. Priyoneel Basu. I'm currently an assistant professor at the Department of Zoology, Dr. Hari Singh Kaur Vishwavidyalaya, Sagar. I'm an INSA Young Scientist Medalist. I completed my PhD from BHU and then went to the University of Calgary, Canada for a postdoctoral research in behavioral neuroscience. So my area of special research interest is circadian rhythms, which is very important in health and disease.
What inspired you to choose this field of study?
Well, actually, circadian rhythms is a very fascinating field, and I was always interested in neuroscience. By coincidence, I was at the end of my master's, I was at a conference on circadian rhythms, and I heard many stalwarts speak on it, and I interacted with Professor Atanu Kumar Pati, who set my interest in circadian rhythms, and then I did my summer training with him and then ended up in this field of science because I started appreciating how incredibly complex it is and how useful it is to human beings and animals.
What technologies or methods did you use in your research?
Well, let's see, that's a variety of methods that I've used. I've used autorhythmometry, I have used circadian wheel running, and then timed injections, then I have used stereotactic surgery, then gene silencing in the brain, and then immunohistochemistry, Western blotting, ELISA, RT-PCR, whole genome sequencing, long-read sequencing, many more, osmotic peristaltic pumps, EEG, ECG, lots of them. Loose clamp recording, electrophysiological recording, lots and lots of techniques. So techniques don't make the research, the idea makes the research.
How does your research impact real-world problems?
Well, actually, my research is completely about real world problems. You see, circadian rhythms evolved in such a way as to enable us to synchronize and time our activities according to the day and night schedule. Now, we have been playing with the natural rhythms a lot through artificial lighting at night, through using cell phones at night, and also through behavioral changes such as shift work and jet lag that we face every day. As a result, our circadian rhythms face disruptions which can lead to very negative health consequences impacting cognition, cancer, cardiac problems, metabolic problems, and a host of other issues. So, my research focuses on which neural pathways underpin circadian rhythms and how can we manipulate those pathways to generate results that help us to better adjust to disruptions of the circadian rhythms that are inevitable in today's paradigms.
Has your work led to any innovations, patents, or publications?
Well, yes, I have filed a patent for an unrelated, I cannot disclose what, and I have a lot of publications which you can look up on Google search if you like, but they are mostly on the neural underpinnings and behavior involved in circadian rhythms and circadian physiology, although I've also worked on cancer, on viral genomes, and many other side topics.
Imagine your body has a built-in 24-hour cycle timer that tells you when to feel sleepy, when to wake up, when to eat, and even when to feel the most alert. This natural timer is called the circadian cycle or the circadian rhythm, and it's also our body's personal daily planner. This cycle regulates behavioral, physiological, and biochemical processes such as sleep-wake cycle, hormone secretion, body temperature, and metabolism.
The Latin word circadian means about a day. It's not just limited to humans. Almost every living organism, from plants and animals to bacteria, has a circadian rhythm. Light exposure, food and meal timing, physical activity, screen time, jet lag, and shift work, all these factors influence circadian rhythms. Maintaining a regular circadian cycle is vital for good sleep quality, stable mood and mental health, efficient metabolism and weight management, enhanced cognitive performance, stronger immunity, and hormonal balance. Why did you focus on the cholinergic forebrain rather than other brain regions?
It's not that I focused on the cholinergic forebrain. Most of my research has been on serotonergic pathways actually, which are quite important because they modulate light input. Now the cholinergic system additionally modulates arousal input. See, that is what we are doing to ourselves. We are looking at light all through the day, all through the evening, and all through the night, very late into the night through artificial lighting, through our cell phones, and then that is causing arousal in our brains because we are getting dopamine fixes from our Facebook posts and our Insta and our Snapchat and WeChat and whatever we have nowadays, and we are enjoying ourselves very late into the night with photic input, doom scrolling, and all of that is affecting our bodies whether we are shift workers or not. It is disrupting our circadian rhythms. That will definitely lead to very serious health consequences. I want to understand what. So that is why I focused on mostly the serotonergic system and a little bit of the acetylcholinergic system. Now at the time when I did this research, a lot was not known about the cholinergic or the serotonergic systems and how they impacted circadian rhythms and what subsets of receptors were involved in these neurotransmitter pathways. So my research has helped establish in some very small ways how we can manipulate these pathways into leading healthier lives, and also helped us understand why there is a lot of individual variation in people and their response to disruptions in circadian physiology.
What makes the suprachiasmatic nuclei such an important part of the brain for timekeeping?
See, the suprachiasmatic nuclei are a group of paired nuclei, 20,000 neurons in total, which fire together. Their oscillation is coupled together, and they have very delicate and very complex both neural rhythms and molecular rhythms that have an approximately 24-hour periodicity. And they have two distinct subpopulations, the core and the shell. The core receives light information and the shell generates the rhythm, the endogenous rhythm that drives almost all physiological parameters, many behavioral parameters, and most of the genes in your body have a circadian correlate. So this is thus a very important driver of many processes that are controlled by the hypothalamus. So it is a central control system and central timekeeping system. It temporarily gates and coordinates behavior so that the best kind of physiological environment is created at the most appropriate moment.
The suprachiasmatic nuclei is a small paired structure that are located in the hypothalamus of the brain. It sits just above the optic chiasm, that's why it is called as the suprachiasmatic nuclei. The suprachiasmatic nuclei is also known as the body's master biological clock. The main function of suprachiasmatic nuclei are: it controls the circadian rhythms of an individual, it controls the digestion, body temperature, sleep-wake cycle, it releases hormone like melatonin, etc.
Suprachiasmatic nuclei is a small group of cells in the hypothalamus that regulates the body's circadian rhythms. It responds to light and dark signals from the environment to synchronize physiological processes with the 24-hour day-night cycle. Suprachiasmatic nuclei. How does it work? Suprachiasmatic nuclei receives light information from the eyes through the retinohypothalamic tract. When there is light, suprachiasmatic nuclei signals the pineal gland to stop melatonin production so that we stay awake. When it is dark, suprachiasmatic nuclei signals the pineal gland to release melatonin so that we feel sleepy. Coordinates the body's internal rhythms. Suprachiasmatic nuclei sends timing signals to brain regions controlling alertness, endocrine system to secrete hormones, digestive system timings, body temperature regulation. The suprachiasmatic nuclei plays a crucial role in maintaining physiological balance and overall health.
What does a phase shift mean in this context?
Okay, so what happens is that when the suprachiasmatic nuclei receive some kind of input, which could be a chemical input, which could be photic input, which could be timed sound, which could be even timed food, they can be reset. Their synchronicity can be reset, especially in the absence of other external cyclic cues. For example, if somebody is in a room that is constantly lit, or if somebody is in a constantly dark room, the endogenous rhythm is expressed. And then if you give a certain timed cue, then this can be reset in such a way that the onsets or offsets of activity or a particular physiological or a molecular parameter can occur at a different time. It can be brought forward or it can be delayed. So this is called a phase shift. For example, if somebody wakes up every day at 6 o'clock and then suddenly moves to an environment where the sun, for example, say England, where the sun is not rising at the original 6 o'clock that the person is used to. Now the sun is rising 5.5 hours later according to the body. Now this requires a little bit of adjustment. If you have a medication or some kind of therapy that shifts the entire body's physiological parameters by 6 hours, then that body is very quickly readjusted to the new time zone.
Sometimes life throws a wrench in that perfect timing. For example, when you have to travel across a different time zone or change your work schedule, in that point of time, how does your body's internal biological clock catch up? That's where the concept of phase shift in circadian philosophy pops in. Phase shift is basically the internal biological clock hitting the reset button to realign itself with the world around it.
It's like adjusting a watch forward or backward so it matches the local time. This shift can happen forward, that is phase advance, going to bed and waking up earlier, or backward, that is phase delay, going to bed and waking up later. Here is a cool example we have. Suppose you decide to start jogging early in the morning. This morning exercise acts like a nudge to your circadian clock, pushing it to run faster, making you feel tired earlier at night and wake up earlier in the morning. Researchers found that consistent morning workouts can advance your internal clock by about 37 minutes. On the flip side, exercising in the evening might delay your clock, making you want to stay up late and sleep in longer. Think of another classic example, light. Early morning sunlight is like a natural alarm clock, resetting your rhythms to the new day. It advances your phase. Meanwhile,
Bright lights late at night tell your clock it's still daytime, causing a delay in your rhythm, making you feel more awake when it's actually the bedtime. This fascinating dance of phase shifts ensures that you are alert when needed and restful when appropriate, helping you to function at your best every day.
Could this cholinergic pathway represent a feedback mechanism for error correction when arousal occurs at atypical circadian times?
That's certainly a very interesting and complex question. The answer would require maybe quite a lot of explanations, but in short, yes, the cholinergic system feeds back onto the suprachiasmatic nuclei, and behavioral arousal can thus influence the suprachiasmatic nuclei to adjust its rhythm in such a way as to respond to the behavioral arousal. And that's why behavioral arousal can reset the circadian rhythm. But again, in some people this does not happen. There are responders and then there are non-responders. So that arousal signaling also depends on a particular subtype of acetylcholinergic receptors. So it's actually a very delicate and complex mechanism.
The mechanism of circadian rhythm is driven by an internal biological clock located in brain's suprachiasmatic nuclei, which is synchronized by external cues like light. The system coordinates the 24-hour cycle of sleep-wake patterns, hormones release, and other physiological processes. The suprachiasmatic nuclei also sends neural and hormonal signals to synchronize peripheral clocks in organs like liver, heart, etc. These clocks also influence metabolic and physiological rhythms like digestion, blood pressure, and hormone secretion. Thus, light indirectly controls the timing of metabolism, alertness, and body temperature.
Molecular mechanism of circadian rhythm. The molecular basis of circadian rhythm is primarily transcription translation feedback loop involving a set of core clock genes. Key proteins such as clock and BMAL1 form a complex that activates the transcription of other genes including period, PER, and cryptochrome cry. These PER and cry proteins then build up and eventually inhibit the clock BMAL1 complex, creating a negative feedback loop that leads to a roughly 24-hour cycle. Light information is captured by the retina. Besides rods and cones, there are intrinsically photosensitive retinal ganglion cells which detect the light using special light-sensitive pigment called melanopsin. The intrinsically photosensitive retinal ganglion cells then send their light signal through a special neural pathway called retinohypothalamic tract. This tract directly connects the retina to the suprachiasmatic nuclei, which is the master clock of the brain. The suprachiasmatic nuclei acts as the central pacemaker that controls the timing of entire body's circadian rhythm. It controls the pineal gland. In daylight, suprachiasmatic nuclei activity inhibits the melatonin production by a multisynaptic pathway. In darkness, the inhibition is removed and the pineal gland releases melatonin, which induces sleep.
Do you think similar cholinergic mechanisms operate in diurnal species, including humans, given the nocturnal model used?
Well, certainly, that's again a complex question. Serotonergic output is different between nocturnals and diurnals, but basically the cholinergic input system is the same and works in much the same way. So yes, it is a parallel study. So yes, it is a study that has parallels in both nocturnal and diurnal systems.
Why were Syrian hamsters chosen as the model organism instead of mice or rats?
Now, that is a very researcher-like question. Yes, we could have chosen mice or rats, but rats are pretty lethargic. Syrian hamsters run wheels very well, and they have bigger brains, so that's a better target. If you are attempting brain surgery, that has to be accurate at the level of 40 microns. So we have to be very, very accurate, and a bigger target is always easier. Plus, their period genes particularly have homologs with humans better than mice. That is what people say. So their responses to light are also nicer than other rodents in terms of. Additionally, phase shifts in Syrian hamsters are better, let us say, than other rodents, and they also run in wheels much better. So wheel running is a very easy and non-invasive way to measure circadian rhythms. So better wheel runners are always better.
A disrupted circadian rhythm is a sleep-wake cycle disorder caused by the misalignment between the body's internal clock and the external environment, leading to fatigue, insomnia, daytime sleepiness, and mood changes.
Major disorders of circadian rhythm disruption. First, delayed sleep phase disorder (DSPD). The person's sleep is delayed by 2 to 3 hours or more. They fall asleep very late, example 2 to 4 am, and wake up late, common in teenagers and young adults. Second, advanced sleep phase disorder (ASPD). The person feels sleepy very early around 6 to 8 pm and wakes up very early 2 to 4 am, common in older adults. Third, irregular sleep-wake rhythm disorder. Sleep and wake times are irregular and fragmented throughout the day and night, seen often in people with neurological disorders like Alzheimer. Fourth, non-24-hour sleep-wake disorder. The sleep-wake cycle is longer than 24 hours and keeps shifting each day, common in blind individuals due to lack of light perception. Fifth, shift work sleep disorder, caused by working night shifts or rotating shifts that go against the body's natural rhythm, leads to fatigue, insomnia, and reduced alertness. Sixth, jet lag disorder happens when traveling across multiple time zones, causing a mismatch between body clock and local time. Symptoms: tiredness, poor sleep, digestive upset. What challenges did you face during your PhD research?
Well, there are always challenges during any PhD at any institute. We faced fund shortages, and then I had to build my PhD lab from the ground up along with my supervisor. It was something that was a challenge as well as an opportunity. I really enjoyed doing that. Funds were always short and equipment was hard to get by, but we managed somehow, and we did some really good research using the equipment that we did have. And we had excellent research ideas, and that's how my PhD was a happy time mostly.
Who has been your biggest influence or role model?
Well, I cannot say any one person's name because there have been so many over the years. J.C. Bose is an inspiration. I want to be like him. And my teachers during my master's influenced me a lot in this field of studies. Of course, my postdoctoral supervisor, Michael Antle, he is somebody that I really look up to, and I wish to be a good scientist like him. So they have influenced my way of thinking, my way of research. Above all, my guru, Shri Shivendu Lahiri Mashe of Varanasi, he has made me who I am, what I am in this way.
What are the most important qualities of a good researcher?
Well, I think every kind of research needs different qualities, but I'll give a non-conventional answer here. The best quality that a researcher can have is curiosity. If you are curious about finding out new things, then you can do good research. Otherwise, it's just incremental. Also, you need diligence. You have to stick to it. You need a certain level of intelligence to make sense of your results. And you need understanding of your particular field a lot. Also, it helps if you can master new techniques easily and apply them. Not only that, I guess what a good researcher really needs is communication skills all of the time, not just for presentations. You need it to write good papers, you need it to present your work. You also need a lot of forward planning, design thinking, and experiential learning.
What advice would you give for handling failures and setbacks?
Well, learn from them and try again. Learn if you still want to pursue that goal or if you want to pursue another goal, but don't give up so easily.
What kind of new researches can be done in the circadian rhythm?
That's again an interesting question. So, circadian physiology is constantly evolving, and there are many new avenues. The molecular basis of circadian rhythms has been always a hot topic, and the correlation between cancer and circadian rhythms is now very well understood, and a lot of research should focus on cancer chronotherapy. And with an alarming increase in artificial light at night and new social paradigms that involve humans waking up later and later at night, there are going to be many health problems, particularly metabolic and cardiac problems. We should look into it really deeply, and we should investigate mechanisms, both neuronal mechanisms and molecular mechanisms, as well as neurogenetic basis for these that tie all of this together. And another important area would be the difference between the central and the peripheral clocks. The clock in the brain, in the suprachiasmatic nuclei, and other peripheral clocks in the liver and the heart, and the asynchrony between them, or the widening gap between them when we expose our bodies to circadian disruption. So there are many such new avenues which are immediately important. We should also try looking for circadian rhythms in many different species, because that tells us how the world is tied together temporally. Perhaps you are doing nothing at all to inconvenience a species other than switching on the lights on a road at the wrong time, and that is impacting their habitat in a photic manner and interrupting their reproduction, and that might be a keystone species. So that is disrupting the whole ecology. So the field of chronotype ecology is very important and has to be investigated now.
Outside of your field, who is your most favorite scientist?
Well, apart from J.C. Bose, I can think of a maverick, Richard Feynman. He got the Nobel Prize, so of course he must be good. And he was also a musician. He was a physicist and solved the Dirac equation, but also was a good biologist. So he did many interesting studies. He had a very sharp scientific mind. He was very thorough, and he was a very good teacher. So he was all of the things that any scientist and teacher should want to be.
Can you share your experience throughout this journey?
Well, there are so many experiences, some of which are shareable and some of which are not. And let's just say the moment when I got my net JRF, immediately after my master's, at the first attempt, that was a defining moment. Then when I started up research and got my first set of results from the equipment, that felt really wonderful. And when I published my first research paper out of my own research work, that felt so nice, because I wrote all of it and I sent it to the reviewers, and I argued with the reviewers. It really felt like I was accomplishing something in the world, in the world of science. And then when I did my first brain surgery, it was great, not for the animal, I guess, but at least for the experiment it was great. We made sure the animal was extremely ethically handled, but it was a really challenging and difficult thing for me to do for the first time, going abroad for the first time, and settling abroad for the first time, living in harsh conditions, climatic conditions, and then doing a lot of research work overnight across many decades, fighting in the COVID war, working 20-hour shifts a day. Fighting in the COVID war, working 20-hour shifts a day, and discovering new mutations in the COVID genome, starting up whole genome sequencing from scratch, and then getting student-level research done at one of the institutes that I worked in, and helping research there, and then coming all the way here by car for the interview, and then 18 hours of driving at a time. Those are also parts of my research journey, because they showed me new places that could be explored in terms of ecology, in terms of human interactions, in terms of human problems. So all of my journey has made me a more understanding scientist rather than just a lab technician.